So hello guys, welcome to the channel. Today we'll be discussing about microbes and human welfare. The topics to be covered in this module or in this chapter will be microbes that are useful in household products, microbes that are useful in industrial products, then the sewage treatment, then microbes in biogas production, and microbes in biocontrol agents, and microbes used in biofertilizers. Now, as we know that microbes are omnipresent, that is they are present everywhere in this world, even in air, water, soil, on your skin, and other places that we cannot even think of, like the extremes of the environment. The they even exist in the extreme environments, like deep inside the geysers or the thermal vents, where the temperature exceeds 100 degrees centigrade. Then meters below the layer of the snow, where the temperature is in minus degree centigrade, and even in the severe acidic environments where no other organism can think of thriving, there the microbes are present. Then let us uh, let us know about what comprise of microbes that we are talking about. The microbes that we are talking about, or we will talk about in this module, will be protozoa, bacteria, fungi, plant and animal viruses, then viroids and prions. In this, viroids and prions are the proteinaceous materials. Proteinaceous materials. Then, to come up with, first we'll start with viruses. And first is the bacteriophage. Bacteriophage, as the name suggests bacteriophage. Bacteria means bacteria and phage means consuming. That means consuming the bacteria. So, in this bacteriophage, we have hexagonal shaped head. Then we have a collar. This is the collar. Then we have a tail piece here, the tail. Then we have the tail plate, which also constitute pins. These are the pins. These are pins. The pins help in attachment and adhering onto the cell surface. Suppose this is the cell surface. With the help of the prongs, the virus lands on the bacteria. These are the pins, and it gets adhered into the cell surface of the bacteria. Like this. Then the the DNA or the genetic material that is present here, the virus sends it into this, and the genetic material gets transferred into the bacteria. These are the functions of the pins and prongs, and this is the bacteriophage. So next, let us discuss about the adenovirus. Adenovirus is a space vehicle shaped virus. Like you might remember the Apollo 11 space vehicle. It resembles that, and it causes respiratory tract infections. Next is the TMV, or the tobacco mosaic virus. It is a compact rod shaped virus. You only need to know this much about the TMV, not much, and it inhabits on the tobacco plant. That is why it has the name of tobacco mosaic virus. Then in microbes, we can cultivate, or we can grow microbes on nutritive media, like bacteria and fungi. Now the bacteria, these both are the petri dish. These are made up of plastic, and it contains agar medium for the bacteria, and for fungus it contains SDA, that is Sabrodes dextrose agar. It is quite higher level for you, but you just need to remember that we can grow bacteria and fungi on the nutritive media, that is agar, and both are agar. Okay. So next, we'll come to the first topic that of today, that is microbes useful in the household products. The microbes that are useful in household products, the most common that we encounter in our day-to-day life is the lactobacillus acidophilus, that is lactic acid bacteria, which helps in conversion of milk to curd, that is curdling of the curdling of milk. The curdling of milk occurs due to the large amount of acids that the bacteria produces, uh, which coagulates and partially digests the milk proteins. We have to remember here that it coagulates and partially digests the milk proteins. It might come as an MCQ where they may ask about do they coagulate and completely digest, or do they necrose or completely digest. These might be the options. So coagulate and partially digest, you must remember to solve the MCQ. Then, as we know how to start curdling of the milk, that is, we first take a spoonful amount of curd and put it into the milk, which acts as a starter for curdling of the milk. Then lactobacillus also increases the vitamin B12 content of the curd, and it also checks on the disease causing bacteria inside our gut, that is our stomach. It prevents the colonization of the gut by other pathogenic bacteria because of its numerous number. Then next to discuss about is dosa and idli. As we might all know how dosa and idli are made, the batter that our mothers prepare, the batter that our mothers prepare, we have seen a large number of bubbles appearing on the batter. The bubbles are due to the CO2 released by the fermentation of the batter. Because of the fermentation and the CO2 release, we see the puffed up appearance of the dough or the batter. This is all because of fermentation, and the organism responsible for this fermentation is Saccharomyces cerevisiae, which is yeast. And because of its property in baking, and most of the bakers use it, it is known as baker's yeast. This is to be remembered. Then the next, many of you might know that toddy is a traditional drink in Telangana and other South Indian states. It is made from sap of the palm, uh, and it is filled in bottles. The it is filled in bottles where the sap turns into toddy. As the day progresses, the amount of alcohol increases with as the fermentation takes place. So it is a beverage, you can say. Then what all other things can we ferment? So traditionally, fish, soybean, and bamboo shoots have been fermented for long and have been consumed as a delicacy in different parts of the world. Then next important topic to discuss is the cheese. Cheese is a very traditional food that we have, and even today uh, it is very famous among the people. So, what are the types of cheese, and how are the types decided? The types are decided based on its texture, flavor, taste, and the organism used in making of the cheese. So, the most important given in the textbook is Swiss cheese. Swiss cheese is made from Propionibacterium cerminae. This bacterium releases a large amount of CO2, which uh, which which leads to the large holes that are present the large holes that are present in this cheese. We can see this much large holes in the cheese, which are due to the large amount of CO2 production. The next is the Roquefort cheese and the Camembert cheese, which are due to Penicillium roqueforti and Camberti respectively. Then we'll see the use of microbes in industrial products like beverages and antibiotics. The beverages and antibiotics are made in very large vessels up to 500 to 1000 liters big vessels known as fermenters. These are used for industrial scale production of the beverages. So, what are beverages? Beverages are the other delicacy drinks that we have. So in beverages, they use yeast, that is Saccharomyces cerevisiae, and because they are used in brewing, we call it as brewer's yeast. So in brewing, there are wine, beer, whiskey, brandy, and rum. And as you can see, the concentration in wine is 4 to 5% of alcohol. In beer is 5 to 10% of alcohol. In whiskey, brandy, and rum, there are 40 to 50% of alcohol, depending upon the brand, which is consumed. So wine and beer are produced without distillation, and whiskey, brandy, and rum are produced with distillation because we need more concentration of alcohol in these drinks. And as you might be knowing till now, that yeast can only produce alcohol concentration up to approximately 13%. Above 13, if the yeast produce above 13%, then they will die because of the alcohol concentration itself. Therefore they need distillation process to increase the alcohol concentration. Then next is antibiotics. Anti means against and bio means life, but this against life is not for us. It is regarding microbes and not us. So going by the definition, antibiotics are the chemical substances that are produced by some microbes and can kill or retard growth of other microbes that are disease causing microbes for us. So going on to the history of antibiotics. So in World War II, in 1945, Alexander Fleming, who was doing research on Staphylococcus, that is Staphylococcus aureus, he found out that when one day when he left his staphylococcal colony unwashed on the petri dish, he found out that on the next day the colony was not allowed to grow on some part of the petri dish due to the growth of Penicillium notatum. And when he went to find what caused this, was because of the antibiotic penicillin that was produced by Penicillium notatum, and hence the penicillin was discovered as the first antibiotic. And as you can see, that it was a chance discovery. It was a chance discovery and not an intentional discovery. So after some time, Ernst Chain and Howard Florey described the full potential of the penicillin, and it led to saving of millions of lives during World War II in the injured soldiers. And all three of them got Nobel Prize in 1945 after the World War happened. So till now, more than 25 antibiotics have been discovered, which have been helping us to cure the patients with plague, whooping cough, diphtheria, and leprosy, which are also known as the dreadful diseases. These are known as dreadful diseases because when untreated, they can lead to 100% mortality rate when untreated. So coming on to the next third topic, that is chemicals, enzymes, and bioactive molecules. First, we'll discuss about the acid producers. You only need to remember 4 of these acid producers. That is Aspergillus niger, produces citric acid. Acidobacter aceti produces acetic acid, as the name suggests on itself. Then Clostridium butylicum. You can see butylicum and butyric acid. You can remember it by this. Then next is Lactobacillus, lactic acid, as you know it already. Next, then yeast produce ethanol, that is brewing or brewer's yeast. We studied it earlier. Next is lipase. Lipase is used in detergents to remove oily stains because oily stains contains lipids. So to break down lipids, we use lipase. The next is proteases and pectinases is used in bottled juices for clarifying of the juice. Next is streptokinase. Streptokinase is also known as clot buster because it relieves the clots in the vessels, large vessels, and it prevents from the myocardial infarction or heart attack. Then cyclosporin A, produced by Trichoderma polysporum. It is a fungus, as you can see, and it is an immunosuppressant. It suppresses the immune system, and it is used in organ transplant patients. Then next are the statins, that is which are produced by Monascus purpureus. It is also an yeast. It is a blood cholesterol lowering agent. It is an important point, blood cholesterol lowering agent, and this streptokinase is known as clot buster. These two are important MCQs. And the statins competitively inhibit the enzyme HMG coreductase. This enzyme which leads to inhibition of the cholesterol synthesis. Then we'll discuss about the sewage treatment. It is an important part. So to start with, what is sewage? Sewage is a municipal wastewater containing human excreta. It contains maximum part, that is maximum part of the sewage is the human excreta, which is drained from the city, and we treat this sewage using heterotrophic microbes, that is anaerobic and aerobic both. In the treatment of this sewage, there are two steps: sewage treatment plant, that is primary or physical step, secondary or biological step. So as you can see in this diagram, the raw sewage is collected from the pipes, and then it is screened and the floating debris are removed in these screens. Then it is passed further down, and this is the first sedimentation part, or it is also known as grit chamber. The grit, like soil and small pebbles, are settled at the bottom, and these are disposed of. The part that remains is passed on to the next tank, that is primary clarifier. In this, the solids that settle down, these are known as the primary sludge, or the primary sludge, and the part that is floating or the clear part is called as effluent or the supernatant. This effluent is then passed onto the aeration tank, where an air compressor constantly sends in oxygen that keeps on keeps on agitating, which increases the growth of aerobic bacteria, which leads to the digestion of the organic matter present in the sewage or in the effluent. This reduces the biochemical oxygen demand of the sewage. So to discuss with what is the biochemical oxygen demand. Biochemical oxygen demand is the amount of oxygen required to oxidize the organic matter present in 1 liter of the sewage. 1 liter of sewage. It is biochemical oxygen demand. Biochemical oxygen demand, it is directly proportional to the organic matter present in the water. Then next, when after the agitation, this fluid is passed on to the next clarifier, that is secondary clarifier. In it the bacterial flocks that are present are degraded, and they are degraded because of the anaerobic bacteria present in it. These anaerobic bacteria clears of the flocks. Flocks are the masses of bacteria associated with fungal filaments to form mesh-like structure. These settle at the bottom and act as the activated sludge, which is pumped back into the aeration tank, which acts as inoculum for the aeration tank. Then after clearing, the secondary effluent is either directly passed into the surface water or is using disinfectant. It is supplied to the city for drinking purposes. As you can see, the anaerobic sludge digesters, as we talked about, they digest the bacteria and fungi, aerobic bacteria and fungi, and which releases gases. These gases are methane, H2S, and CO2, which are also known as biogas. The large amount of sewage that has been produced in the cities nowadays is continuously increasing. So the Ministry of Environment and Forest, that is MOEF, has introduced the Ganga Action Plan and Yamuna Action Plan to reduce the pollution of the major rivers, that is Ganga and Yamuna. Next, as we have seen the biogas, we'll further discuss about the biogas production. Biogas constitutes of mainly methane. Methane is the main component of the biogas. Then second is the hydrogen sulfide, CO2 and H2. Biogas production occurs due to the cellulosic degradation of products by methanogens. Methanogens are methanobacterium, which are present in the rumen of the cattle. Rumen is a part of the cattle, and it also occurs in the anaerobic sludge as we have seen earlier, which leads to formation of the biogas. So it is also known as gobar gas because of association with the cattle. This here you can see the biogas plant. In the biogas plant, we put water, wastewater feed, that is the slurry, cattle dung plus water. We mix it and we put it as a slurry. These are the 10 to 15 feet deep dug tanks. The slurry goes down into the digesters. In these digesters, the degradation of the cellulose degradation occurs and the gases are released. After the release of the gases, the lid of the digesters are covered with a floatable gas holder, which adjusts its position according to the gas produced. And there is a primary valve here which controls the release of the gas into the pipelines. It is further connected to the pipeline gas holder. Then the used up slurry is collected into this manual bed, and this is used in agriculture as a fertilizer. Note, the biogas production was developed first in India. India was the first country to introduce the biogas production, which was due to the common effort of Indian Agricultural Research Institute, that is IRI, and Khadi and Village Industries Commission, that is KVIC. These are the two important institutes responsible for the development of biogas production in India. This might come as MCQ, IRI, KVIC, and where was it first produced?